Please welcome.
Thank you for this introduction, and thanks to all the organizers for giving me the chance to stand up here today. So before I jump into my research project, which is about awakening a sleeping carboxylase, I would like to give you a brief background about what we are doing in the lab of Tobias Erb. So we all know that our lifestyle is leading to an imbalance in the CO2 household in the environment. And while the nature is able to fix and release CO2 at about the same rate, our human activities emit way more CO2 than we are able to fix through industrial processes. And CO2, we all know, is a potent greenhouse gas, but what we tend to forget is that CO2 is actually a carbon source that is freely available out there, and we just need to find a way to grab this molecule and turn it into something valuable. And that's what we're trying to do in our laboratory. So we're trying to look for novel carboxylases that could do this job. And the first step towards this aim was the discovery of the reductive carboxylation, which is the most efficient principle of carboxylation known up to date. And we have studied this enzyme very extensively in our lab. And we basically could identify 4 different residues that are essential to bind the CO2 molecule within the active site. And by knowing this, basically we call it the CO2 binding motif, we could understand these enzymes, and this motif is very well conserved. So, but one carboxylation reaction is of course not enough to turn CO2 into some valuable product. So what we try to do in our lab is to design and establish novel synthetic CO2 fixation pathways that don't exist like this in nature. And what you see here is our prime example, and you see that it involves 2 of these very efficient carboxylation reactions. And in the net equation, the cycle can fix 2 molecules of CO2 and release 1 molecule of glyoxylate. And to just give you the key points for this cycle, it contains 17 different enzymes. They come from 9 different organisms across all 3 domains of life. And it even contains 3 enzymes that we engineered towards the required reaction. So we purify all these enzymes and basically combine them into 1 Eppendorf tube, and what they reach is a CO2 fixation rate of 5 nanomole per minute and milligram enzyme. And this is comparable to running the CBB cycle in vitro. And I said it relies on this very efficient carboxylation reaction exclusively. All right, so while we are trying to continuously optimize this cycle, we also try to find novel carboxylation pathways, to engineer them, and therefore we need to find novel carboxylases. And what you see in this colorful slide here is basically the workflow towards finding these novel carboxylases. So I told you we have this very efficient carboxylation reaction at hand, and by studying it, we could identify the CO2 binding motif that is conserved in the protein sequence. So with this on hand, we could go out and screen other protein sequences if they maybe contain the same CO2 binding motif. And indeed, we did find some candidates, but these candidates were not initially efficient carboxylases, so they had to undergo several design, test, build cycles to turn them into proper carboxylases. And I want to talk about one example that we found through this approach and that we engineered to become a carboxylase. This example is the propionyl-CoA synthase. And as you can see from the structure here, it comprises 3 different domains: ligase, dehydratase, and reductase. And I can mention at this point that the reductase domain is actually structurally and phylogenetically very close to these ECR enzymes. And we could find the CO2 binding motif in this reductase domain. So far, PCS has been described to perform the overall reaction from 3-hydroxypropionate to form the reduced product, propionyl-CoA. But we suggested now that with the CO2 binding pocket, it might be able to also carboxylate the intermediate acryl-CoA to form methylmalonyl-CoA. So we tested that in a first very straightforward experiment, which is we just let the overall reaction run in presence or absence of bicarbonate. And what you see this is that if we have bicarbonate in the assay, we can detect a small peak by LCMS that corresponds to this carboxylated product, methyl malonyl-CoA. However, independent of bicarbonate, the major peak is always the reduced product propionyl-CoA. So you might think now when you look at these peak sizes, wow, PCS is a miserable carboxylase. And yeah, you are right. But this small potential actually motivated us to go out and try to engineer this enzyme towards proper carboxylation. All right, and we employed two different strategies to engineer this enzyme. So on the one hand, we tried to improve the carboxylation activity, while on the other hand, we tried to disfavor the reduction activity. And what you see is the active site of the PCS reductase domain. And if you can recall the active site of the ECR, it basically looks identical. And the 4 important residues are also conserved. So why is PCS not a better carboxylase? We had a closer look, and we identified this aspartate over here. So it's always an aspartate in all the PCS sequences. And our idea was that this aspartate might actually lead to a misorientation of this very important asparagine residue over here, in a way that at the end, the carbonyl group points towards the CO2 molecule. Of course, that would destabilize this interaction. So the idea was, we mutate this aspartate to a serine, how it is often found in the ECR sequences, and this mutation might then correct for this misorientation, improving the CO2 binding. So, and on the other hand, what you see is the entry site where the intermediate acryl-CoA enters the active site of the reductase. So in here you see the CO2 molecule and the binding pocket. And what you can see is that this entry site is a pretty big hole, and it probably would allow free diffusion of water into the active site. The water can then displace the CO2 molecule and lead to reduction rather than carboxylation. So we again compared it to the ECR structure, and we tackled this residue over here. It's a threonine in our homologue that we are working with. However, it's always conserved to be a methionine in the ECR structure, sequences. So, bringing in this mutation, threonine to methionine, you see that we dramatically decrease the size of this entry site. And this might now actually prevent free access of water to the active site. So we heterologously expressed these variants and tried to characterize them. And that's what you see in this slide. So it's basically a Michaelis-Menten plot. In black, you see the methyl malonyl-CoA generation rate over the different CO2 concentrations. And in gray, you see the carboxylation efficiency in this bar plot. And you see, if we measure the wild type, we cannot saturate the enzyme with CO2. So the binding, we cannot determine any binding parameter for CO2 in this variant. However, if we now introduce this positive design element, we can saturate the enzyme with CO2, and we can determine a Km for CO2. So this indeed showed that we improved CO2 binding within the active site. And on the other hand, we implemented this negative design element, and you see it didn't change anything about the CO2 binding. However, it already improved the carboxylation efficiency compared to the wild type. And if we now combine these two mutations into one variant, we get to the following picture. So we have this improved CO2 binding, we can measure a parameter, and the carboxylation efficiency even increases up to 95% at very high CO2 concentrations. All right, you might wonder now at this point, why do we need such a carboxylating PCS enzyme? And PCS, we could show in previous studies, is actually very unique. It forms this so-called reaction chamber in each of the chains. And this reaction chamber is like a perfectly enclosed sphere, basically. And if you look into the inside of this reaction chamber, it connects all 3 active sites of the three domains with each other. And we could show that PCS is sequestering this intermediate acryl-CoA inside this reaction chamber. So it never lets it diffuse out into the bulk solvent. And we actually have one synthetic CO2 fixation cycle that we tried to establish in our lab that uses exactly this reaction sequence from the hydroxypropionate to methylmalonyl-CoA. And you can imagine, like, having an enzyme that sequesters this acryl-CoA would be of a very big advantage, because acryl-CoA is a very reactive intermediate, and if it would react with other intermediates of the cycle, it could lead to dead-end products. And with this, I want to just give you a very quick summary. So we could identify PCS as a sleeping carboxylase because it has this CO2 binding motif, but it carboxylates only at a very small efficiency. And through rational design, we could wake up this carboxylation activity, and we could now probably use this, this designed carboxylase in our synthetic CO2 fixation pathways. And with this, I want to thank the group at the JGI who are providing us with many genes for these carboxylases or potential carboxylases. And also thank you to Soichi and Hassan from SLAC who collaborated with our lab to identify and describe this CO2 binding motif structurally.
Thank you very much. Questions? I actually thought the really great work. So evolution-wise, so this ECR complex, the ECR enzymes, are they evolved from these like propionyl-CoA like reducing enzymes? You think?
I think it actually happened the other way around.
Oh.
So like that, the PCS fused together from 3 different enzymes, and the last domain was one of these CCR. Yeah, that's our hypothesis. But of course, evolution is always really tricky to prove or to disprove.
Right.
Yeah.
Have any questions?
There is one.
Oh, yeah.
Mm-hmm. I mean, the affinity is actually not higher compared to the single mutant, so the Km was still the same. But you are right, the efficiency of carboxylation was already super high at this low carboxylation. And of course, the lowest CO2 concentration that we measured in these assays was 4 millimolar, and that's a totally feasible value that can be reached in an organism. And of course, the PCS that we studied comes from a heterologous organism, so it produces propionyl-CoA, and the next step is an ATP-dependent carboxylation of propionyl-CoA to methylmalonyl-CoA. And if we could now implement this carboxylating PCS, it would skip this ATP-dependent step and would be like a computational advantage.